Thank you. I'll try and make this quick as well so we can catch up. So Venezuelan equine encephalitis virus belongs to the family Tocoviridae, and it's an alphavirus. One of the reasons that we're particularly interested in this virus is that it's a category B select agent, which means that it has weaponisation potential, and it's overlapped with priority pathogen for the CDC and the USDA. It's a positive single-stranded sense RNA genome, and it replicates in the virus in the cellular cytoplasm. But interestingly, the capsid protein of this virus, which I'm going to call VIV for the rest of my talk, does some very interesting things to host cell nuclear transport. So the VIV capsid protein has been shown to be a major pathogenicity factor. It has multiple functions, including packaging of the viral genome. It has protease activity. It's a host cell transcriptional inhibitor, and it's the main reason for the extensive CPE seen with VIV infection. So the inhibition of cellular transcription is independent of its protease activity and its RNA binding domain, and it seems to be dependent on a short amino terminal peptide. And this capsid protein is mostly found resident at the nuclear pore complex, which is embedded in the nuclear envelope, and there it's been shown to be a potent inhibitor of cellular nuclear cytoplasmic trafficking. So nuclear transport in the cell is a very tightly regulated process. So in a general sense, proteins that need to go into the nucleus contain a nuclear localisation signal, or an NLS, and these are recognised by members of the important superfamily of proteins, which can either be recognised by important alpha as part of the important alpha-beta heterodimer, or via important beta directly. And in either case, important beta mediates binding to the nuclear pore complex and translocation through into the nucleus, where binding to RAN-GTP dissociates the complex and releases the cargo. Nuclear export is a very analogous process where other homologs of the important beta family known as exportins, of which the most well-known is CREM1, bind to cargo-containing proteins containing a nuclear export signal, or NES. And these happen in a trimeric complex in association with RAN-GTP, and it's basically the same process in reverse, where the important beta, or in this case CREM1, mediates translocation through the NPC to the cytoplasm, where hydrolysis of the RAN-GTP to GDP causes the cargo to be released. In the case of infection with VIV, however, the capsid protein interferes with this process in several ways. Firstly, it can bind to the important alpha-beta heterodimer, as well as the CREM1 complex, forming a tetrameric complex, which is believed to sit within the nuclear pore complex. And this inhibits both cellular export as well as nuclear import. And it's in this case by physically blocking up the pore, as well as mopping up the nuclear import proteins, that capsid is expected to inhibit cellular nuclear transport. And the virus does this because the virus doesn't require the nucleus for viral replication, but it's shutting down nuclear transport in order to prevent host cell viral inhibition. So while this is a very nice theory, and there is some really good evidence to show that this tetrameric complex occurs, the actual nuclear trafficking of the capsid protein has been very poorly studied. And the reason that we want to look at this is because actually these interactions become very important targets for viral inhibition or inhibitors. However, it's only through understanding these interactions that we can actually begin to start inhibiting them. So what I'm showing here, is a protein-protein binding assay that we use in our lab called AlphaScreen. It's a bead-based binding assay, which I won't go into. And what I'm depicting here is binding of a very well-known nuclear transport cargo, nuclear import cargo, the T antigen protein from the SV40 virus. And I'm showing you this because this is a typical profile of what a protein that uses the important alpha-beta pathway looks like. So we get very strong binding, shown here in green, to the important alpha-beta heterodimer. Important beta alone, shown in red, shows no binding. And important alpha on its own shows minimal binding, but this is because important alpha undergoes, when it's in the absence of important beta, it auto-inhibits itself. So when we use a form of important alpha known as delta IBB that doesn't have that auto-inhibitory domain, you can see that we recover all of the activity that's found in the important alpha-beta heterodimer. And this is very typical of an important alpha-beta cargo because it's the important alpha protein that binds to the typical NLS. In the case of the capsid protein, however, we see something very different. So you can see here in green, we get very strong binding to the important alpha-beta protein. However, for alpha, beta and the delta IBB, we see about 50% residual activity. And what this tells us is that capsid protein is likely binding to both alpha and to beta. So this could indicate two things, either that it has both an active important alpha-beta pathway as well as an important beta pathway, so two separate pathways, or it could be that it's binding to all of these proteins in a non-productive manner, where it's just binding them but it isn't actually mediating transport. So we wanted to investigate this further, so we utilised our in-house in vitro nuclear transport assay. So in this assay, we mechanically perforate HTC cells to isolate the nuclei. And on top of this, we can add a fluorescent protein. So in this case, we could use our capsid protein. We can add anything we want. We add an ATP regenerating system because it's an active process. We add back in exogenous cytosol, so this is our source of importance. And importantly, we can add in antibodies to the various different importance or inhibitors, and this can help us to tease apart what pathways are being utilised. We then look at these through confocal laser scanning microscopy, and over time we can get an accumulation of the fluorescent protein within the isolated nuclei, and we can quantitate this. And for this we use a measure we call FNC, which is the nuclear to cytoplasmic fluorescence ratio, and you can see over time, as we watch these nuclei, the fluorescent protein accumulates rapidly inside the intact nuclei, and then we can get kinetics of transport from this measurement as well as maximal accumulation. So when we utilise this assay on capsid, we can find lots of things. So firstly, when you just put capsid protein into the assay, we get a very rapid and impressive accumulation of capsid within the nuclei. When we include in the assay this detergent called CHAPS, which is a very well-known permealiser of the nuclear envelope. So basically it just punches large holes in the nuclear envelope, and therefore anything can just flood into the nucleus. We can see that we remove all accumulation of the protein. So this tells us that capsid accumulation is very dependent on an intact nuclear envelope, and therefore a nuclear pore complex process is going on. The other assays that we can do is, so this one here where we add BSA, basically we don't put the cytoplasm into this sample, and we replace the missing protein with just BSA. And as you can see, the accumulation here is greatly reduced. So this indicates that this process does require cytoplasmic components, most likely importance. And we can also add antibodies. So if we add antibody to one of the important betas, important beta 2, we basically see no difference. So this indicates to us that important beta 2 is not needed for capsid nuclear accumulation. When we look at the most common importance used here, so in this case important alpha 2 and beta 1, we can see that antibodies to both of them greatly reduce the capsid import activity, with important beta having the greatest effect. And this is in contrast to that protein that I showed you earlier, T antigen, where in fact it's the important alpha antibody that has the greatest effect, with important beta having only residual effect. And the reason for this is, again, that we think capsid is having a slightly different transport process than important beta, but this assay shows us that there is actually productive transport occurring both through important alpha but also directly through important beta. So it looks likely that capsid actually has more than one nuclear transport pathway, which is not uncommon for viral proteins. So I've said that. So the next thing is to see whether this actually has an effect on capsid accumulation in whole cells rather than just in the isolated nuclei. So here I'm looking at infected cells. So these cells have been infected with a laboratory strain of VIV, and we've used siRNA to deplete either important alpha, beta or both. And you can see that in the place of just a scrambled siRNA, there's really just normal accumulation of capsid. It's quite nuclear. However, when we add siRNA to important alpha and beta, basically we see a great reduction in the FNC of capsid, indicating that capsid's no longer able to get into the nucleus. It's not very clear on this screen, but you can really see here that these nuclei are much darker than the ones up here. And this occurs with both alpha and beta singly, with beta having slightly more of an effect, and also with the two of them combined. So this tells us that alpha and beta are playing a role in infection, in getting capsid into the nucleus and most likely into the nuclear pore. We can do the same thing here looking at nuclear export. which we weren't able to do in our isolated cells. And here we can deplete the nuclear export inhibitor, nuclear export in CREM1, and we see a very dramatic increase in nuclear transport. And so obviously, if you're inhibiting nuclear export, things get stuck inside the nucleus. So you can see here that now the capsid protein is pretty much solely contained in the nuclei as opposed to being more cytoplasmically distributed in the negative sense. So both import and export inhibitors seem to affect capsid nuclear accumulation. So we can do the same thing. In our lab, we've been working for a number of years now on generating small molecule, very specific inhibitors for nuclear export and import. And we looked at a few of these in the case of VEEV. Now, these molecules that I'm showing today weren't targeted against VEEV, but they do have activity because VEEV uses a very similar pathway to the virus that they were generated against. So I'm showing two here today, mifepristone and ivermectin. Mifepristone seems to be a viral-specific nuclear inhibitor. It seems to bind to the viral protein and prevents it from binding to important alpha-beta, whereas ivermectin is what we call a broad-spectrum important inhibitor. It actually binds to the important alpha-beta proteins and prevents them from binding to cargo. And because of that, ivermectin also inhibits host cell nuclear transport. So it's a little bit toxic, but I'm just using it here as an example. So you can see here when we add either of these proteins, we see the same thing. We see a significant decrease in capsid accumulation. And this shows us that blocking these pathways is actually a viable way to inhibit capsid from getting into the nucleus. And then we can do the same thing with nuclear export. So there's a very well-known inhibitor of CREM1 called leptomycin B, which is very specific for CREM1. And when we add leptomycin B, we see a very dramatic increase in capsid nuclear accumulation, indicating that blocking nuclear export of capsid is also viable with a small molecule inhibitor. Now that's all very well and good, but does it actually have an effect on the cells and the way that they respond to the virus? And the first thing that we noticed was that when you infect cells with VEEV, it's very toxic to the cells. The cells rapidly die. And this is a luminescence assay measuring cell viability. So you can see that with infection, we see a 50% decrease. This is about 18 hours after infection. However, when we add either our nuclear import inhibitors or our nuclear export inhibitors, and these are at very low dose, we see a dramatic increase in the amount of cell survival. And this tells us that nuclear transport inhibitors increase cell survival following VEEV infection. So they seem to be having a positive effect on the pathogenicity of the virus. And basically, the reason for this is that the inhibitors actually inhibit VEEV replication. So when we look 16 to 24 hours post-infection, particularly with mifepristone, we don't see as much of a significant effect with ivermectin. However, we have to use ivermectin at very, very low doses in cell culture. It's extremely toxic in cell culture. It's much less toxic in whole animals for some reason. It's actually a licensed compound. But in cell culture, you can't use it above, 500 nanomolar. So we see only very small effect with ivermectin. However, leptomycin B has a very dramatic effect on the viral replication, decreasing viral titers very significantly. And all of these can be repeated. So TC83 is the lab strain of VEEV, which is the non-virulent strain used for vaccination of horses. However, when we repeat this with Trinidad donkey, which is the completely virulent wild native strain of the virus, we see the same effects. So a significant decrease of about a log for mifepristone and several log for leptomycin B, suggesting that import and export inhibitors definitely can inhibit viral replication. And lastly, we can carry this forward into looking at other related New World alpha viruses. So here I'm showing Eastern equine encephalitis virus and Western equine encephalitis virus, both of which are very important pathogens in their own right, perhaps even more so than Venezuelan virus. And here I'm just showing nuclear export inhibitors, so Lep B is leptomycin B that I showed you before. And this is a whole panel of nuclear export inhibitors that we've generated as a part of this project. KPT301 is the inactive variant, so you can see that it has no effect, whereas all of these other export inhibitors have a dramatic effect on the replication of these two viruses, indicating that they must behave in a similar fashion as we demonstrated with VEEV. So in conclusion, we can show you that VEEV capsid undergoes active nuclear transport, presumably via multiple important mediated pathways, and that inhibiting these pathways both alters capsid nuclear accumulation in both transfected and infected cells, and that inhibitors of these pathways increases cell survival following infection and reduces viral titer, and that there are similar mechanisms observed in the related viruses. And so we're obviously following this through our hit to lead process, and we've actually got some much more potent inhibitors now that follow a similar mechanism but have a much more dramatic effect on the virus. So this work has been really a cross-continental collaboration between a few groups at my university at Monash, with some groups at George Mason University, as well as Karyopharm Therapeutics, who make the nuclear export inhibitors, and Schechter Computational Solutions have been doing some molecular modelling of the work, and it's all been funded mostly through a large grant from the Defence Threat Reduction Agency. So thank you.